Hi! In this video, we will show you a method that uses flow cytometry to assess cell disruption of microalgae. Flow cytometry is used to count and analyze the shape, size, and properties of single cells within a mixed population. In a flow cytometer, a sample containing cells enters the equipment and is mixed with a saline solution. Cells move then to a narrow channel and are disposed in a fine line, right before entering the interrogation point. At this site, each cell absorbs the light of the laser and the scattered fluorescence is captured by the detectors. The signal is then processed with the computer and displayed into beautiful plots. In this video, we will show you a protocol that uses flow cytometry to analyze the cell membrane integrity after a culture being subject to disruption with a high-pressure homogenizer. So let's start! For this method we will need the dye Cytox green, which is impermeant to live cells and therefore used to assess membrane integrity, the phosphate buffer saline, abbreviated as PBS, the microalgae culture. In this method we used Tetradesmus obliquus grown in piggery wastewater, and of course the flow cytometer. We used the Cytoflex from Coulter Beckman, equipped with an argon blue laser and 5 detectors. So we first adjusted the flow cytometry parameters to the Tetradesmus obliquus by comparing a live with a dead culture that we previously boiled in water for 60 minutes. By analyzing these two, we were able to identify and define the gates of the different cell populations. After passing the culture through the high-pressure homogenizer, samples were collected and diluted with PBS to read samples from 100 to 150 events per second. Now pick your working stock solution of Cytox green and stain your PBS diluted samples. You should always optimize the concentration of Cytox because it varies depending on the cell concentration, species, growth stage or cultivation media. In this method, we used Cytox at 1.8 micromolar and incubated samples for 25 minutes in the dark, since Cytox can suffer photodegradation. As you may know, cell size can be assessed using the forward scatter detector, shortly abbreviated FSC, and cell granularity or complexity using the side scatter detector, abbreviated SSC. Cytox green binds with high affinity to nucleic acids and emits fluorescence at 523 nanometers. Therefore, we use the FL1 detector, which collects light in the green region. This methodology can distinguish 3 different cell subpopulations. Cells that were not stained with Cytox and are located in the upper region of the forward and side scatter detectors have the cell membrane intact, so we name them intact cells. On the other hand, cells that were stained with Cytox and therefore have high FL1 fluorescence got a compromised cell membrane. However, if they appear also in the forward and side scatter detectors of the intact cells gate, they still maintain their size and internal content, so we name this permeabilized cells. Cell debris caused by severe disruption is located at lower forward and side scatter fluorescence, so we name these disrupted cells. We hope that this knowledge is now at your side, so happy flow analysis! Get to know us at greencollab.com. Thank you for watching.